We do put these on our website after they're over, so people that weren't able to join us today live can do so later at their convenience. So with that, anything else about your background, Jeanne, that you'd like to share with the group? I don't have your bio in front of me. I apologize.
Well, you said the most important thing, which is I'm from Wisconsin. And a proud graduate of the University of Wisconsin at Madison.
And the mascot is?
Bucky Badger.
Bucky Badger, of
course. As everyone in the universe knows, we thought.
Well, I bet you don't know my alma mater's mascot. I'm from Stetson University, DeLand, Florida.
Hat?
Hatter. The Hatters, you got it.
Okay, I get partial credit.
Yeah, hey, I'll give you full credit. And Jared Westbrook, PhD, is from University of Florida, and those are the Gators.
Yeah.
So anyway, Jared, anything you want to say before we get started? Kind of why we decided to ask Jeanne to do this? Maybe you can share with the group. And Jared is our Director of Science for TACF, if you haven't had the pleasure to meet him.
So one of the things we're doing in TACF is we are, in fact, tomorrow, inoculating trees with the chestnut blight fungus, the Americans, the Chinese, and F1 trees, the hybrids between the two species. And we're looking at, in the stems, which genes are expressed in response to the fungus, and comparing the resistant susceptible species to develop some hypotheses about what are the mechanisms of resistance. And part of that is also looking at the metabolites that are expressed in the stem, and that might be important for, especially early on in the infection. We want to know, why is it that the fungus tends to grow faster in the American trees and be suppressed in the Chinese trees? What is that X factor? So our use with this metabolite profiling is more in understanding the basics of how does the resistance work in the Chinese chestnut tree. But Jeanne is going to present on how this could be used also in a practical breeding program sense, understanding, can we select for ash trees that are resistant to the emerald ash borer by making a quick metabolite profile of these trees without having to do all of the difficult phenotyping for resistance to emerald ash borer. So there's multiple practical uses and basic uses of this technology, and Jeanne will kind of talk about both of that. So we'll leave it to Jeanne.
Ah, wonderful. I can talk now.
You
well,
can.
Hello.
Jared and I will go off screen, but if you need us, just holler.
If anything goes wrong, I'll immediately call on you. Deal? Okay. Hello, I am very pleased to be invited by TACF to give this talk to you today. I'll tell you some about plant metabolomics and give you a little context and a little background about what is that and what is it good for. We've had omics around for about 20 years. High-tech omics. The first omic that we set our sights on was, wouldn't it be great if we could sequence all the DNA in an organism? That would be awesome. 20 years ago, it cost $100 million to do the human genome. That was the estimate. Well, now we're very good at it. We have high throughput sequencing. So when we talk about genomics, we mean that, the ability to rapidly sequence DNA from any organism on Earth. Okay. The next omics is transcriptomics. That DNA carries information. That information has to be translated into another molecule called RNA. And then the RNA has to be transcribed. The RNA has to be transcribed from DNA. Then proteins are translated from the RNA. So all the RNAs that are actually transcribed from the DNA, that set of stuff is called the transcriptome. And now we have the capacity to sequence the transcriptome too. So that's the transcriptome. Transcriptome makes, for the most part, proteins, okay? And now we have the technology to identify most of those proteins fairly rapidly. So that process is called the proteome. And then finally, we now have really wonderful instruments to identify every chemical species, small molecules that are made in an organism. The proteins are usually enzymes, and the enzymes generate these small molecules. So when we were first doing omics, we thought, well, the complexity will be found in the genome, then the transcriptome will be easier, and the proteome will be easier yet, and the metabolome should be easier yet. So we're looking forward to that. And it turns out, now that we can do all these omics, that it's exactly the reverse. In terms of complexity, the metabolome, the thousands and tens of thousands of little molecules that are made, that's much more complex than the proteome, the transcriptome, or the genome. And just for all us folks who work on plants, the plant metabolome is much more complex than the animal metabolome. So there, animal people. All right. Omics is nice, but why would metabolomics, a metabolomics approach, make sense for the TACF? Right, first, let's look at an integrated workflow for breeding for biotic agent resistance. If you're going to use no technologies at all, this integrated workflow is still what you would use in a traditional breeding program. The little boxes specify an activity. The letters above the boxes specify where traditional methods are used, robotics potentially, omics, tissue culture transformation, and possibly CRISPR. Okay. So over here in the orange, this is when the focus is on the agent. You don't start a process like this unless you recognize, oh, there is a biotic agent that's causing damage and death. So we usually focus on this end of the process first. But once you get into it, identification of species, biotypes, races, if we're interested in saving a forest tree, say chestnut, from blight or ink disease, the first thing we want to do, oops, I knew I'd do that. First thing we want to do is identify the sources of resistance, right? If not in the species, then in the related species. And that's what TACF did many, many years ago. They initiated a breeding program with Chinese chestnut. Now that we have the omics, we can better integrate what we know about the biotic agent and what we know about the tree and the host to better identify good phenotypes, more rapidly identify good phenotypes, and even ultimately identify the fine details of plant-agent interactions so that we might be able to actually edit the genome to get resistance much more quickly and effectively. Okay, so what part of this workflow is relevant for the TACF? Well, the goal of TACF is self-sustaining, durably resistant populations with sufficient recovery of genetic diversity to ensure that the restored chestnuts are going to be here 200 years from now, when none of us will be here to see it. And the biggest impact that you can make in terms of ensuring that that goal is met is right here in phenotypic selection. Okay, even if you have a transgene or you can use CRISPR, ultimately it always comes back to you're going to make some crosses, you're going to pick out some trees, so the phenotypic selection part of the process is never dispensed with. Okay, all right. Now, so what are the most cost-effective omics tools for phenotypic selection. Well, the most cost-effective one, of course, is direct phenotypic selection. If you call it phenotypic selection or phenotyping, no one will fund you. But if you call it phenomics, well, then you just might get funding to be able to do it in an academic context. But this is more important than anything. So, but phenomics takes a while. It's labor intensive. You do it when it's really hot and horrible, or when it's raining. It's difficult and it's time consuming. What's the omics as near as possible to this direct measure? As near as possible to this direct measure, almost as good as phenotyping, is the metabolome. Those small molecules that ultimately determine the phenotype. They are the last step before the phenotype that we can see. So what is the metabolome anyway? All right. Plants, and trees included, make a dazzling array of secondary metabolites. Biochemists like to draw out these elaborate pathways here, and these are the main categories of secondary metabolites, little molecules that are used primarily for defense. The tree can't run away. It's got to defend itself against fungi, against bacteria, against insects, against herbivores that are chomping on it. Biochemical defense is what plants use to do this. Plants also use these small molecules for signaling to their endophytes. Plants have a microbiome, same as us, for signaling to each other. Hey, I'm under attack here. Or for signaling to insects that can come in and gobble up other insects that are eating them. Okay, so these secondary metabolites are legion. There are 21,000 known plant alkaloids. There are 15,000 known, approximately, isoprenoids that plants synthesize. And these are not simple molecules, okay? And there are many, many flavonoids and complex flavonoids. Now, the target specificity of some of these secondary metabolites can be very broad. There are some that a plant will synthesize in response to any stress whatsoever. And those aren't really helpful to us when we're trying to incorporate resistance to a specific disease. What we want is those metabolites that are very, very narrow. We know that Chinese chestnut, a good healthy Chinese chestnut, is probably not going to die of blight. And yet an American chestnut is going to die of blight. And what's the difference there? We want to know what is that subset of metabolites that makes that difference. Well, and why are there differences anyway? Any plant has an intrinsic genetic capacity, hard-coded in the DNA, to produce a range of metabolites, 10 to 20,000 different metabolites in each individual. But what you actually see at any given time is subject to extreme variation. It can be dependent on species. It can be different between individuals within species. It can be different between tissue types within an individual, and it can be different depending on the developmental state within tissue types. And of course, as is always the case with complex traits, environment strongly influences the actual realized host capacity. And a good example of this is Chinese chestnut. If Chinese chestnut is subject to a very severe drought, it can die of blight. It dies of the blight because it is simply unable to marshal its defenses. It's rather like a human who has severe diabetes and high blood pressure, heart disease. That person's much more likely to die of COVID-19. Their immune system's not particularly in good shape. Okay, so we want narrow target specificity, and we got zillions of things to look at. How do we do that? Oh, here is just an example of how tissue specificity can be. Camptotheca acuminata is the Chinese happy tree. This is this compound that you see is camptothecin. Camptothecin is a powerful anti-tumor agent, okay? There's not much of it in a fully mature leaf, but there's a lot of it in immature leaves. So you have to know something. You have to, when you first start a metabolomics project, it really helps if you know something about your system. All this analytical chemistry is done by my collaborator, A. Daniel Jones, a professor and director of the mass spec facility at Michigan State. Okay, so here's a summation of the known pathways for the synthesis of secondary plant metabolites. So this is the simplified version. Yet we know from the metabolomics that we've done in the last 4 or 5 years that 80 to 90% of plant secondary metabolites are unknown compounds. We can isolate them. We know we have distinct chemical species, but we don't know what they are. But that doesn't mean we can't use them to accomplish a goal. Okay, so how do analytical chemists do this? This actually works much better than genomics or transcriptomics. In this technology, we take the samples, we extract them in some sort of combination of solvents, and we run them through a series of analytical devices. In the cartoon here, we show liquid chromatography and mass spec, usually abbreviated LCMS. This technology is capable of not only isolating down to the individual compound every metabolite in the sample, but it also reports back to you what their molecular mass is. This is the molecular mass of camptothecin here. It gives you a visual, a chromatograph. It gives you estimated, very accurate masses for peaks at the level of sensitivity that you have declared. And it does a lot of other things. This system, the specialized equipment and the specialized software and the analytical chemist to run it can do this. This is not something you do in your garage. This is something that requires a very specialized lab. So we feel fortunate to have a collaborator at Michigan State, not very far away from us. It is also important to have statistical advice because associating these compounds with some meaningful biological phenotype requires that you understand what the statistical analyses, how they're used in this sort of world in metabolomics analysis. And provided that you understand that, or at least can be advised about it, you can interpret your metabolome. Okay, here's a typical chromatograph. The chromatograph is produced for the purposes of the humans looking at it. This isn't actually how the machines work. So this is one extract of one Chinese happy tree, immature leaves sampled at one time of day, okay, in one type of solvent mix in the positive ion mode. So you see all these peaks. And these are real peaks. These are real chemical species. And you see camptothecin is this peak here. It took a lot of work and tens of millions of dollars to determine that the anti-tumor agent is this one and what it looks like, okay? But what you see in this chromatograph isn't everything. If you look at that chromatograph closely, on a 10x scale, you see a lot more peaks. And what an analytical chemist does then is you dilute your sample and you run it through again to pick up the small peaks, because sometimes the metabolite or the group of metabolites where the action is are found in these very small peaks. So these aren't broken up molecules. These are individual chemical species. So when you do this, It is very typical in any plant metabolomic study to find 1,000 metabolites in your tissue of interest. Okay, that's quite a typical thing, and 80 to 90% of them will not have determined structures. And how do we know that? Because chemists are very organized, and everything that has a determined structure is in chemical databases somewhere with a known mass. So part of the specialized software, the specialized software says, okay, here's all our masses, what's out there that's known? And then all that is delivered back to you in a specialized facility. Okay, goody. Well, what do you need to know to take advantage of this wonderful technology? Well, it helps. What you do beforehand determines how it ends. Do you have a scientific question? You know, this technology is not very good for going fishing because you'll get a lot of results, but you won't know what it means. Can you answer the scientific question with the host material available? Have you done preliminary studies? What do you know about your system? What do you know about how the metabolites respond in your system? Do you have direct access to the biological samples at the right time? That's really critical. And now you have to decide how you're going to store them, how you're going to extract them, and if the metabolites are stable. If you're doing something that involves leaf volatiles, for example, these compounds may not be stable, so you have to do something different to look at leaf volatile metabolites than to look at, say, a metabolite that is active in a stem canker. And you have to be aware that no single solvent is going to dissolve all the metabolites, so that's another reason you need analytical chemists to give you some advice about this. You have to choose which one of the wonderful machines in analytical chemistry to use and which combination. You choose that ahead of time. What kind of data collection method are you going to use? These systems are all automated or semi-automated, but all this has to be decided in advance. How you're going to process the data. Are you going to do standard database searches? And then a step that a lot of people haven't gotten around to yet, validating your results. That doesn't come in the analytical chemistry facility. That comes from you. You do all this, you'll be able to interpret your data. If you don't do this, you will find a lot of things, but you won't know what it means. Okay. So how to make metabolomics work for you in any situation whatsoever. Understand the sources of phenotypic variation and design your experimental approach to detect the sources of variation or to avoid it. 2, know what your goal is and stick to it. So here are 2 goals that TACF is interested in. The host breeding program. Now, this is just my interpretation of what the goals are, so whatever Lisa says trumps me. And there's a genetic mechanisms goal. You work out the genetic mechanisms so that you can do direct intervention in an applied breeding program. If I understand very clearly what the genetic mechanisms of blight resistance are, I might be able to use CRISPR to edit the genome, but I cannot use CRISPR until I've done my homework and worked out what those genetic mechanisms really are. Okay, so I'm going to give you an example of how this can actually work in the field. I have a type A goal in my Emerald Ash Borer program. The type A goal is using metabolomics to increase the efficiency of an applied breeding program. We're not primarily interested in working out the molecular biology of the interaction. So our primary goal is to develop green ash with enough resistance to EAB, to maintain genetic diversity, self-sustaining populations, and so on. Our secondary goal is development of a field test kit that could tell us which of those trees in the forest that are still alive after EAB comes through, which of them are actually killing the larvae. Because there are some that don't kill any larvae. They are just lucky. And those trees don't have anything to give their progeny. Their progeny aren't going to be resistant. Fast forward, what's our progress towards the goal? This project is successful. We have full stem progeny with larval kill as good as Manchurian ash. Okay, as good as Manchurian ash. 20 years ago, people would have said this is impossible. So we set out to do that which is impossible, because that makes me mad when someone says something's impossible. How about our secondary goal? We have identified a set of 5 to 9 metabolites, depending on genetic background, that correctly identify 80% of the trees with high larval kill. Now, when you look at this, you might say, well, that's not very good, but it's actually spectacularly good for metabolomics analysis. So, did we need metabolomics to achieve our primary goal? No. But what putting the metabolomics in there that did is enable us to get money so that we could keep this program alive. Okay, metabolomics sounds a lot more exciting than phenotyping. And that allowed us to pursue my lab's secondary goal, which is development of a field kit. Okay. My collaborators in this project are Jennifer Cook and Therese Poland, both of USDA Forest Service. This project has taken 26 years, but we started from a dead zero. We started from nothing. So think about when the TACF was first founded. 1984, maybe before that, 1974. I'm not really sure. I'm sure Lisa and Jared can tell me. That's when we started. And in 26 years, we have success. Okay, in 26 years, we are close to being able to use metabolomics to make that breeding program more efficient and actually restore the species to the forest. I think that's pretty good in terms of trees. Now, what about TACF? TACF has both type A and type B goals. You have an applied breeding program, but you also want to elucidate or work out the precise genetic mechanisms of host pathogen interactions. And when that is your goal, and if you can marshal the funds to do it, you have a system where you understand how the canker works, you know it's susceptible, you know it's resistant, then metabolomics can be an excellent tool to help you to understand what these mechanisms are, so that some of the more technologies like CRISPR then actually become viable. Okay, I always end a talk like this with my standard slide of best practices. If you're going to do this, if you're going to do any long-term breeding program, and you're going to bring in omics, which tends to be a little spendy, you need professional guidance. Not any program has all of these professional individuals except the private sector. But no university program has all of these individuals. We have to go out and find people to bring all the individuals together in a collaborative effort. Okay. 2 clear goals, regularly reviewed, and 3 long-term commitment. Without a long-term commitment, you can't achieve either a type A goal or a type B goal. You have to stick with it for the long term. Your tree lives many, many years longer than you do. It's important to respect that and stick with your goals. All right, I'm done talking now, and I would be happy to take questions.
Thank you, Jeanne. That was wonderful. I really learned a lot being a non-scientist, but play one on TV sometimes. We have some questions in the Q&A, and a comment from Hill, who said, "Tell Jeanne I said, good job." He had to go at noon and do a lecture like you do at 1 o'clock. So
yeah, well, thank you, Hill. I'll tell him later.
Okay. The first question is, where in Wisconsin are you from? From Frank.
Ah, I was born in Portage, Wisconsin, in Columbia County, and my mother's entire family, all of them, live in Dane County, Wisconsin, where I spent most of my life until 20 years ago. And I am very proud to be from Wisconsin, and I eat cheese.
Great. Okay. Let's see. My wife, and another one from Frank, my wife and I spent 4 hours collecting samples of original American chestnuts for DNA sampling. As far as I know, we did everything as requested. However, we were told later they were not good. What happened?
Well, it depends. Were you collecting leaves or twigs?
Can you put that in the chat, Frank? Leaves or twigs?
I can answer the question.
Okay, cool.
Yes, sure.
What we had, this is for the landscape genomics study, where we're characterizing the diversity of American chestnut, the genetic diversity across the landscape.
Yes.
And the first year for the collection, I don't know when you did the collection, but it's very important that the leaves are young if you collect leaves, because they have a lot of, the older leaves have a lot of starch and other secondary metabolites that interferes with the DNA extraction. And so at Virginia Tech, they were able to optimize the DNA extraction somewhat, but if any of these older leaves, if they were like leaves that have been on the tree for like, let's say months, then we couldn't get good DNA out of that. So there's metabolites in there, but that's what interfered with the DNA extraction.
Yes, when leaves are good for high quality, abundant DNA, but I tell people, I mean, I don't want to see leaves collected later than first week in July. Leaves don't grow anymore after they fully expand, and as a leaf ages, it gets the equivalent of leaf wrinkles. It gets beat up, secondary metabolites build up, it undergoes a lot of oxidative stress, so the DNA is just not good in older leaves. Even though they might look green and great to you, they're not so great when it comes to DNA extraction. Did that answer your question?
We'll just assume so unless they do a follow-up in the chat.
All right.
This is from Bruce Levine. For the non-biochemists, can you please explain the difference between metabolites and secondary metabolites?
Yes, this is a distinction that biochemists make. You could call glucose a metabolite. Okay, when a plant photosynthesizes in its chloroplast, it uses energy from light to synthesize glucose. So that is a metabolite, but we don't call the biochemicals that go into fundamental energy metabolism as secondary metabolites. Secondary metabolites are those metabolites that go through a long process of construction, and they are for a specific purpose other than just to burn fuel to keep the organism alive. Okay, so these tend to be in plants. They tend to be alkaloids and terpenoids, which are synthesized only for defense or signaling. So that's the difference. Although in some cases, the distinction gets a little gray.
Great, thank you. From Mark Stotz. Hi, Mark. Do you see an analytical method like LC/MS as a replacement or an adjunct to field methods like the small stem or large stem assays?
It could be a replacement if you do the necessary homework to understand, to be able to interpret what those metabolites are telling you. It's nowhere near that now because the really carefully designed experiments to tell you that information haven't been done. But ultimately, it could be. There's no easy way to develop a metabolite assay for the field unless you go through a number of years of really carefully designed field work, okay? But we're doing that in my lab because our phenotyping is really painful and difficult. It involves putting eggs on, you have to dissect every stem. It takes a lot of labor and a lot of time. So we have an incentive. Your phenotypic assay is, it's not that it's easy, but it's a little less onerous.
Great, thanks. Okay, from Fred, fascinating primer for me, thank you. I have no scientific background, so please excuse my ignorance. That being said, I see a lot of parallels from this line of research with the current moonshot push for C19 research. My question, would there be any possible application of the research tool folding at home to help with this? I'm not familiar with folding at home. Are you, Jared, or you and Jeanne?
I don't understand what folding at home means. Someone needs to tell me that.
I'm not sure either. Can you, Fred, can you, in maybe another comment, you tell us what that means, and I'll go on to another question. Again, from Bruce Levine, is it helpful to know the metabolite you are looking for before you start, in other words, from other pre-metabolomic research? Is it better not to have preconceived ideas and just see what the trees tell you?
It's, well, it depends upon how relevant that research is. There are very fast and fairly cheap targeted metabolomics methods. If you're looking for a specific compound or compounds that might be related to that compound. Now, the risk you take is that you've already made up your mind that this is the thing you want, whereas that might not be the case. I mean, there are very large differences across species, for example. There are very large differences even between individuals within the same species for certain metabolites. So it's a matter of how good was the previous research, all right? How convincing is it? How carefully did they do their field work? But it's a targeted metabolomics is a fairly low bar, so it's worth considering. Somewhat high risk of missing the mark, but it's not that expensive.
Okay, great. Fred did put in the chat room a link to foldingathome.org, and Tim X says that's a citizen scientist protein folding program.
Oh, yeah, where citizen scientists can donate their computational power to
it's a mass processing tool.
Yes, to attack the issue of, given this amino acid sequence, how is this protein going to fold up? Okay, and that's a very difficult computational problem. It might be nearly intractable were it not for having hundreds of thousands of people's computers working on it at the same time.
Okay, hopefully that answered your question, Fred. From Greg, what if, and it is possible that, blight resistance is not caused by one or more secondary metabolites?
That's entirely possible. So when you do this, you need to have an experimental design that helps you sort out, I'm going to see a lot of metabolites. Do the metabolites have anything to do with the resistance response? The technology cannot tell you that. It's your experimental design and your your materials that you have in the field where the resistance response is already known. Okay, that's why the experimental design has to be thought through carefully. Otherwise, you won't know.
Okay. From Alan Nichols, hey Alan, has any of this technology been used to find and or develop American elm or American beech that could be used for restoration?
It has not been used on American elm. And part of the reason for that is that there appears to be, at least in many regions of the country, in the forest, American elm is regenerating somewhat well all by itself. Now, this is probably because those trees have been rigorously selected for being able to deal with Dutch elm disease. For, what was the second one? Beech bark disease? Beech bark disease, my colleague Jennifer Cook also works on the beech bark disease. And it turns out that the inheritance of beech bark disease resistance is fairly simple. So there hasn't been much incentive to use metabolomics on that problem.
Okay, great. From Stacy Clark. Hey, Stacy, she was our Chestnut Chat speaker 2 weeks ago. Hi, Jeanne. How do you go about determining which metabolites would be important in identifying pathogen resistance to blight? And would these metabolites change as the trees age or change in their environmental conditions, like going from open sun to shaded conditions?
Of course they can. So I can tell you how we did it in green ash, all right? We developed a system in which we put the eggs on the trees 3 years after we graft them, because then they are big enough and they are about 8 feet tall, and all this work can be done in the greenhouse. We can, and we know that the rating we get is similar to the rating outside because we have grafted clones of the original lingering ash tree outside. So one of the reasons it's taken 26 years is that the first 15 were done verifying that the experimental conditions that we have chosen are good for accounting for sources of environmental variance. Okay, but once you do that, then you need some sort of population structure to sort out the signal from the noise. So we use full SIB families from lingering ash by lingering ash and lingering ash by a classic susceptible cultivar. We also know that the emerald ash borer resistance response is induced. It's not constitutive. How do we know that? Because we have uninfested controls. So to do these experiments takes a lot of work, and you work in structured populations. Okay, it's difficult to do these. It's just as a landscape approach, because you have different genetic backgrounds, which will give you different metabolic profiles, sampled at different times, at different developmental states. So you have to figure out a way to control for all that noise and still get a valid rating, and that can take a while to figure that out.
Okay, great. Another ash-related question from Mike Alcott, who is a great scientist in his own right. Given that there might be 1,000 metabolites in a tissue sample, were there any particular methods or tricks even that you use to identify the 5 to 9 metabolites that identify ash trees with high larval kill?
We did the 1,000 metabolites on every single sample, but we also phenotyped in our method. The tree is actually phenotyped by dissection at the same time that we take the tissue for metabolites. So we have trees of known phenotype, and ultimately we performed our contrasts in segregating full SIB families and multiple segregating full SIB families. So you compare the trees that kill the most larvae in one full SIB family with the trees that kill the fewest, okay? And you compare that to individuals from that same full SIB family who are uninfested. And you do that across full SIB families. When you do that and you do something called a discriminant analysis on all 1,000 metabolites for every individual, the question you ask is, okay, are there a set of metabolites that correctly classify my high and low larval kill trees? And that's how you find them, okay? And then you go to the next full SIB family and you say, hey, is it the same 5 metabolites or is there just overlap? And that's why I say 5 to 9, because there's overlap, but it's not complete. But that's all right, we expected that. So in this way, you can hone in on which metabolites matter. But the power to do that honing in is strictly a function of the experimental design we used, and the fact that we phenotype and take the tissue at the same time. So we know what that tree's phenotype is. That enables us to do something called a discriminant analysis. And that is how most metabolomics projects are done. So you don't take all the metabolites and perform some sort of averaging. No. You use a method that looks at all the metabolites in all the samples, but you have already classified the samples ahead of time using your phenotyping. And that's what enables you to do that. I hope I don't have a chalkboard or anything. I don't know if
You're doing great. You're doing great.
Right.
We'll hear back if people need clarification.
All right.
Here's another ash question from Richard. Were the compounds associated with resistance to the emerald ash borer characterized chemically? Are they toxic to the emerald ash borer?
Well, we have 5 to 9 compounds. If you look at all 9, okay, the set of 9, none of them are in the existing chemical databases. These are terpenoid alkaloid compounds with wicked ring structures. To elucidate the structure of one would take an analytical chemist several years. So we haven't done that yet because we don't have the funding to do that. And we know from our analyses that it's not just one. There seems to be a need for a group of them at the same time. So lacking the funds to do that, we haven't pursued that. We only know the general chemical class, terpenoid alkaloids, which would surprise no plant biochemist anywhere. But we can still develop a diagnostic test without knowing exactly what these structures are. It would be good if these structures could be elucidated, though, because I suspect resistance to stem borers, the mechanisms of resistance to stem borers is an unknown country. People tend not to study this because it's hard. Getting at the phenotype is hard, but I'm hoping that people will do it. 10 years from now, we'll know more.
Great, thank you. From Anna Conrad. Hey, Anna. When developing a phenotypic tool based on plant metabolites, do you think constitutive or induced, or both, boy, Anna, you're really challenging my pronunciation, are going to be important? Also, how many years of field trials do you think would be required to get a robust metabolite-based tool? Two-part question.
Okay, first, constitutive versus induced. If a plant synthesized all the secondary metabolites of which it is capable, especially long-lived forest trees that have a dazzling array of these things, I'm not sure it would be able ever to have any seeds. This takes a lot of photosynthesis. Okay. As a result, and based on all the research I did in the private sector a long time ago on stalk borers, I would be very surprised if any of these are constitutive. That means, you know, the tree has to be challenged with the pathogen or the pest. Okay, it simply costs too much to have all your guns blazing all of the time. It's much more efficient to say, oh, all right, the bad guys are coming over the hill over there, and just point one gun at them to give you a grim image. Okay, so constitutive meaning the metabolites are always expressed, I don't think so. Now, intrinsic genetic capacity is different. If we could work back from the metabolites all the way back to what's different here, what parts of the genome determine that these metabolites are actually going to happen, that would be great, because then we could do a DNA test. But that takes a while, because these systems are at least the systems we work in, these are not simple systems. If you're lucky and you happen to work in a simple system, then you have a much better shot at getting back to the DNA and then being able to do a simple DNA test that you can do on a little seedling this big.
And Anna also said, really enjoyed your talk.
Oh, thank you, Anna.
Yvonne has a comment and then a question. We in
Okay.
Rhode Island are hoping that the very new small leaves we collected last month gave good DNA. We noticed that some trees are still putting out very small number of brand new leaves in mid-July. Fingers crossed. And then she asks, if a pathogen has multiple avenues of attack on your organism, can these techniques discern multiple metabolites involved in resistance?
Yes, but think about it. You've got to generate and execute an experimental design to do that. We strongly suspect, for example, that when an ash tree is under attack, an ash tree that's mounting some kind of chemical defense, most of them don't. Most of them just let themselves be eaten up and they die. But an ash tree that's mounting a chemical defense, we suspect that those trees are also signaling through leaf volatiles. So we're doing a leaf volatile metabolome study to see if we can detect by leaf volatiles which trees are actually effectively killing the larvae. And it might be that some trees don't kill the larvae in the stem because they mobilize metabolites in the wrong tissue at the wrong time. So to do that, you've got to do a very careful multiple tissue, multiple time point study. And those studies are really worth doing. They take time, they take money, and oftentimes funding agencies aren't particularly impressed. But hey, we got to fight the good fight and do it anyway.
Amen. Okay, from an anonymous attendee, does the metabolomic change with the addition of the oxalic acid oxidase gene, or OXO, in American chestnut tree change? So does it change it? Please excuse my unsophistication, as this is an excellent and educational presentation.
Well, we don't know that yet, but I would hope that TACF, Jared and the other scientist he's working with would do that. Okay.
That's been,
yes, Jared.
It's the oxalate oxidase gene insertion. Sorry, my dog is running in here. Does not significantly change the metabolite expression compared to the wild type trees. And that was like looked at, and actually in the USDA petition, they compared the wild type trees to the trees that have oxalate oxidase in them, and they have very similar metabolic profiles. And it's actually more similar between the transgenic trees and the wild trees as compared to the backcross trees compared to the wild trees, because the backcross trees have a lot of different Chinese chestnut genes in them, and that influences the metabolites that are in the trees. So
yes, it does.
These trees are behaving more like American chestnut.
And that in the OXO gene, you have a case of understanding how your organism works can lead you to a solution. Just blight is a necrotroph. It kills the tissue, then it eats it, and it kills it with oxalic acid. So oxalic acid oxidase destroys the oxalic acid. So in theory, and that would be the only thing that would happen, and it certainly looks as if that this is a very targeted approach based on a fundamental understanding of how this fungus kills.
And if I can just interject related to that, a very important week for the research out of ESF, SUNY's Environmental Science and Forestry School, Bill Powell and his team. It has been posted on the Federal Register, their petition to deregulate the transgenic tree, the Darling 58. Jules posted the public comment period webpage for instructions and links directly to make positive comments, hopefully, if you'd like to participate. We are really pulling out all the stops to try to let the our constituencies know that this is live now, and we'd love to let the USDA know how much we need this tree. So it's right there in the comments. All right, we're doing pretty well. We're about 12:25. We have 6 questions and a couple of comments left. Can someone from TACF, Jared, explain as to whether this approach would help the TACF efforts? Take it away, Jared.
So in terms of making selections like what Jeanne was doing with ash trees, we can do that with our phenotyping method, and it's actually a lot easier to just inoculate a seedling and get a canker than to extract metabolites and spend a bunch of time analyzing the metabolites. So I think from a practical point of view, in our breeding program, our phenotyping that we do with inoculation and just looking at the severity of the cankers is adequate. But in terms of understanding the mechanisms of resistance in Chinese chestnut so that we could potentially engineer or add in additional resistance genes beyond the oxidase gene, for example, from Chinese chestnut, we need to understand what are the mechanisms that Chinese chestnut is using to resist the blight. And it's more complex than just simply oxalate detoxification. There's probably other metabolites that the Chinese chestnut is secreting that is suppressing the fungal growth. So what we're doing now is the first step is just comparing the Chinese and the Americans to see what is expressed in the stems in response to chestnut blight. A lot of that, we probably won't know what those metabolites are, but we may be able to identify some highly expressed metabolites that are present in Chinese chestnut in response to the blight that are absent in American chestnut, and those would be candidates for further characterizing. And if we know what they are, then we can say that maybe we could go back to the genes and understand what genes cause those metabolites to be expressed. So that's what we're doing now. Is we're actually going to be working with Oak Ridge National Lab. Hill Craddock is doing the inoculations of these trees this weekend, and we're going to be sending these samples to be analyzed with Oak Ridge to characterize the metabolite profiles in the American and the Chinese in response to blight. So we are pursuing this line of research, but it's mostly to understand the mechanisms of resistance and less in the practical realm of selecting trees that have resistance. Yeah.
Thanks, Jared. As you can see, everyone, we have lots of collaborators. We couldn't do this all by ourselves. And Steve Barilovitz, Jared has a comment. Jared, this sounds like a lot more breeding. And just so you all know, Steve is the chair of our Science and Technology Committee. He's a 20+ year active volunteer and board member in TACF. And Steve was the one who recruited Jeanne to this talk. So thank you, Steve. Let's go back to a couple more questions. Would infecting a tree with a symbiotic fungus induce the formation of protective secondary metabolites? Sounds like a PhD to me.
In science, you never say never. I'd say, well, that's an interesting question. Let's do it and see what happens. Of course, then one has to convince a funding agency that this is the best thing since sliced bread and doorknobs. But, you know, there are some of us are very, very good at it. That would not be me, but there are other of my colleagues who are. So I don't know of a single study that has that focus, but I think it would be good. There's plants, especially forest trees. Well, forest trees tend to have ectomycorrhizae, mycorrhizae that grow in tight association with the roots. But there are also endomycorrhizae, fungi, and endobacteria that get inside and we believe perform a protective function. So perhaps one hypothesis is that the plant doesn't need to attempt to metabolize its metabolic defenses if its endophyte is doing that job for it for the small price of a little carbohydrate, some sugars every now and then. Sugars are something plants have a lot of, so that's a cheap price to pay. But that's just one theory.
Cool. Okay. Jim Tate, does the growth of the omics studies incorporate natural history and conservation specialists better than old-fashioned biochem and genetics were doing?
Well, my first impulse is to ask the questioner how old he is, because in the old days, before I was an undergraduate, people did do that. And then as we got into DNA and nucleic acids, our approaches got much more specialized, and we tended to not pay attention to quantitative genetics and quality control and analytical biochemists so much, because people were very focused on that the DNA is going to tell us everything. The DNA sequence will tell us everything. Your point is well taken. You do need to incorporate this into your experimental designs, into your discussions of what should we do to investigate this problem, into your scientific question. I mean, it's always possible that, when a tree is in the forest, hanging out there on its own with a whole community of other trees, that its metabolic responses may be different, perhaps may be more finely tuned. And the only way to investigate that is to take our metabolics out into forest plots and deploy them there. And that's part 3 of our grand scheme here, not funded yet. But yes, that needs to be considered, because your ultimate goal is restoration of self-sustaining populations where nobody's going to be there to take care of the tree. So you've got to know, you really do need to know how that tree and its descendants are going to perform, or at least, and my colleague Stacy Clark knows this, you've got to take the trees that you think are going to perform well and put them in a forest setting to actually see what they're going to do. There's no replacement for that.
Jim Tate posed that question, and he shared some demographics. He's a PhD. He's 80 plus, 80 something, and a retired science advisor to the Secretary of the Interior. Wow. Welcome.
I'd say, listen to this man. He knows a few things that many people have forgotten.
Amen. All right, we have a 3-fer from Frank up in Wisconsin. First of all, a couple comments. He says he's from Mouston, but prefers fresh cheese curds, beets cheese any day. I don't know if you want to respond to that, Jeanne.
I can. Fresh cheese curds. Okay. You got your fresh cheese curds, fanatics, provided that they're fresh, like they were in the cow yesterday, okay? And you got your battered and deep fat fried cheese curds, fan, and I love both kinds.
Well done. Very, very diplomatic there, Jeanne. Another one from Frank. This may be off topic a bit, but I was wondering how current logging practices affects the genetics of a forest. Apparently in the far past, selective harvesting large trees reduced the size of the remaining trees.
There have been many studies on this, many studies in applied forestry on this. This practice is called high grading. So you just take out the best stuff and you leave the rest. And the concept is that you're degrading the gene pool, perhaps. The problem is you need to run these studies for a long time. And during that time, land use changes, the forest composition changes, you have invasive species that come in. So it's really hard to know what has changed. I know of some studies in black walnut that when you just look at the general population genetics of high grading, not specific genes that control timber type, but general population genetics with just a handful of genetic markers, you find that high grading doesn't make all that much difference, but that's a very weak kind of evidence. It's just very difficult to get this information because of the time it takes before you know anything. There may be something to it. one thing we can all agree on is coming in and clear cutting everything over huge swaths of the landscape is probably not a good idea.
Okay, a couple more from Frank, and then a final comment from Jim, and I think we've cleared out the Q&A box.
Okay.
Great questions, everybody. Frank asks, is there any indication that ash trees can call pileated woodpeckers to eat boar?
Well, the pileated woodpeckers really like them.
Right.
Okay. There have been some studies that have shown, while pileated woodpeckers just have a good time eating emerald ash borer, it does not seem to impact the mortality rate. Okay, that it would be good if that could happen. We have no evidence that the trees can call the woodpeckers. All right, I would be surprised if that were the case, because emerald ash borer isn't exotic. Okay, it's native to China, and it was only detected in the late 90s, early 2000s in Windsor and Detroit. So that's not much time for that to have evolved. But what I would do, if it were easier to do, is go to China and see if there are any analogous types of birds, types of woodpeckers that do that, and study that interaction.
Great, and then a couple of comments, Wisconsin oriented. Cheese curds straight out of the vat is best, again from Frank. And Jim Tate said, thank you for the kind response. By the way, I was born in Mouston, Wisconsin. I hope I'm pronouncing that correctly, in the driftless area.
Oh, beautiful place. The latest, the Wisconsinian extension of the Laurentide ice sheet didn't cover all parts of Wisconsin, and the driftless area is just one of the most spectacularly beautiful places on earth.
That's wonderful. Jeanne, excellent workshop and presentation. Thank you so much. Jared, thank you for your contributions. Steve, thank you for recruiting Jeanne. All of you, this is really great to have these Chestnut chats and engage our community. We're still doing good. TACF strong. Finished our spring appeal, raised as much money as last year, amazingly enough, in this really challenging time. Thank you for all your support and all you do to keep this mission going. We're in it for the long haul, as Jeanne said, long-term commitment. So thank you all very much. Have a great weekend.